Tone. I have the pleasure actually of introducing to you Sidney Brenner, who's a geneticist and winner of the Nobel Prize in Physiology and Medicine 2002. Continues to be one of the world's leading pioneers in genetics and molecular biology. He established the existence of messenger RNA with Francis Crick back in 1961, discovered the triplet nature of the code of protein translation, which provided critical insights into the nature of the genetic code. His recent work involving new ways to analyze gene sequences has led to fresh insights into vertebrate evolution. He is a Fellow of the Royal Society, a Foreign Associate of the National Academy of Sciences, a recipient of the Lasker Award in Medical Research, and currently Distinguished Fellow at the Salk Institute. We learned last evening that as a mentor and master, he is demanding, but the results seem to be well worth it. He told us last evening that 5 of his postdocs are also Nobel laureates. I asked if he had any open slots.
Too late.
Yeah, he said that's what he told me. It's too late. We are delighted to have you with us today, Sidney.
Yeah, well, I thought I'd begin at the beginning because I think there is a lot of confusion about this synthetic biology. So let me begin by saying that the most important thing to understand about the living world is that it is the only complex natural system which contains an internal description of itself. And I can illustrate this by a story which I think is important to understand. I once attended a long and boring discussion by a Buddhist priest. And at the end of this, someone said, "What is the Buddhist definition of life?" And he said that many Buddhists believe that everything is alive. Rivers are alive, mountains are alive. I interrupted him and I said, "Mountains are not alive." And he said, "How do you know?" I said, "You can't clone a mountain," which is a title I've patented for the book. You can't clone a mountain because the mountain does not contain this internal description, which of course are the genes, because that's what you hand down from generation to generation. That is what changes, that is what differentiates one organism from another. So DNA by itself is useless. It'll probably just fall to pieces in a laboratory. You must be able to express what is in the DNA, and if you want to do anything with it, you need to copy it. That's organisms have learnt how to do. So if we were to ask, how should we treat all of this, we have to ask, well, what are we synthesizing? So let me now go to another area of synthetic, which is chemistry. Now, we know that in chemistry, you can start to study natural products, and you can try to find out their structure by degradation. That's the way all chemists. But the proof that you've got the right structure is that you can make it. You can synthesize it. Now, in that case, these people have to put elements together in a way which is not the way they were put together inside the natural product. And so they learn an alternative way of making that, which of course is then deployed in making entirely novel compounds. So I think there is a very important place for what I'll call synthesizing DNA or synthesizing genes, which is to prove that you really know how it works. Today we live in a period where we think we can describe everything in the world at atomic resolution, so we don't have to bother to understand or prove anything. But if we are going to understand living things, we need to prove we know how this gene is turned on or what disease does. It's not enough to say, to give just a description. That's not science. That's something else. So I believe that one has to start with this fundamental thing, that of the DNA, which then has to be interpreted by a living system in order to do anything. Has no function as such. It has promise, but it only has promise by itself. So if you take that point of view, I would like to add one other very general thing. About 60 years ago, John von Neumann, a very great man, whom everybody knows from other things in this country, was concerned with how you would build self-reproducing machines. And he produced a very interesting set of theorems, if you like, that there were conditions for constructing such self-reproducing machines. I read his article in 1950, and I was astounded by this because later it turned out he predicted the whole of the DNA and the structure of living systems. So it is one thing, and in fact, this was unknown to all the people working who seem to be more influenced by Schrödinger, though Schrödinger can be shown to have been wrong. You see, Schrödinger said the chromosomes contain the program for the organism and the means to execute them. But they do not contain the means. They contain a description of the means. You have to read that. So we have to distinguish in inheriting things between two things. One, the proposition of continuity. You first have to make something using the machinery of one, and then you have to, using the description, so you must already have the reader. So it's a moot point whether if you synthesized a whole genome and did a really, say, critical Ventarian experiment, whether it would actually work. Because if the genes there were incompatible with being read by the machinery of the recipient, you would not get anything. His experiment, I tried to do his experiment many years ago, just by standard mating, which was the question is whether if you put E. coli, the whole chromosome of E. coli into Salmonella, it was a favorite question. We used to ask students, what do you think the outcome will be? And everybody thought it would be sort of in between, you see. The safe answer is wrong, because then you find it doesn't work, because there's one failure in the reading mechanism, because you can't get that out of Salmonella DNA until you have it. Okay, so there should be one gene that you would then have to put an E. coli promoter there, so you can read it, so it could go on and read itself. So there are very strong conditions for moving DNA extraneously. So I think we really need to see this. And just in that last discussion we had, I think the nightmare incident, which I think we could think about, is the suicide infector. Gives himself smallpox, travels to a country, goes around to movie houses, and can create an epidemic in today when nobody is vaccinated. And I think we should look at the natural way in which infections moved, like the SARS epidemic. And you can see that those which are the products of nature's genetic engineering, simply because you have a lot of people and ducks or people and other animals living together, viruses will exchange things, and you create novel pathogens. The other thing I would like to say that you need to have firmly in perspective is the following. It used to be argued, especially at Asilomar, that the dangerous thing was to cross natural genetic barriers. In other words, to put DNA. We always considered cloning orange DNA and duck DNA together, what that might be, thought of. It resulted in a completely distorted view of pathogenicity. The view was the more pathogenic the organism, the higher the level of containment. So in a sense, lions should be cloned at a much higher level of containment than pussycats. Clearly lions are more dangerous than pussycats. But that took no account into the reconstitution of the pathogenicity. In order to get an effect, as I said, the DNA is useless. You must reconstitute the actual pathogenic mechanism. And so I think when we do this, we need to keep all of these things in parallel. Because what Venter has done is he's a forger. He's created counterfeit genomes. That's exactly what he's done. He's copied another genome. He's also, like most forgers, he actually has signed the forgery. You see? So we know it's him. But this is not a evil thing. He's synthesized nothing. He's created a counterfeit which just does what nature does. So he's proved that nature can be emulated in that way, just by copying. And I think that's fine. We knew it anyway from the normal one. So I believe that there is still a very important area of misunderstanding of what the technology can do. And even in the example, if I may mention it, of the bioterrorist who orders a pathogenic sequence from this, he'll have to amplify it in some way. Which means he'll have to protect himself from it. Or else he'd be like those Irish bombers who would blow themselves up at the wrong time in the streets of Birmingham. But he will have to have much more than just a garage in order to create a real public hazard. So I think we'd really have to know, as someone said, who is ordering the DNA. Now, I'd like to just finish off by saying that really, what are the visions for all of this technology that can come? So we've heard of all the applications. None of them are really novel in the sense that they hadn't been already thought of with ordinary genetic engineering. I think that, personally, that the most important thing we can understand is the discovery of how devices work at the molecular level. We have three classes of devices in the world, or gadgets, if you like. You heard about all of this. They're just making little gadgets here. So there are devices. There are the devices that come out of synthetic organic chemistry. There are the devices that come out of what's called nanotechnology. And there are the devices that can come out of biological systems. Now, if you look at the character of all of these, they have different modes of specification. They have different modes of fabrication. And the area that is completely missing is how we put these all together to make things that are of use and of function in the real world. So how can we employ the great specificity of biological recognition at the molecular level when we don't have we only have very crude methods of putting that into a device, like making an array and so on. We can't read from a sequence directly into a computer. We have to go through a whole lot of other machinery. I think that that's an area of technology. In fact, when I started a new lab recently, which you have to be under 30 to enter, and in which you have to know what you want to do. I'm not going to tell them what they want to do. So this is a lab to train real scientists. And so I had to find a name for it. So since for the last 50 years, all we've done is, append the name molecular to everything. So we have molecular gynecology, molecular medicine, molecular what you like. I believe it's even molecular psychiatry, which sounds like something we should use for the DNA of pathogenic sequences. However, so I thought, what about molecular engineering? I'd never heard this before. So just for desultory, I actually looked for it in one of these literature abstracts. There's a paper written in 1981, I think, by Drexler, the same man who's, propagated nanotechnology, in which everything synthetic biology says, and more, and of course less, because the technology didn't actually exist, is written there in that paper. Of course, it inspired him to do the things in nanotechnology as well. So I think that presaged, and I think that is the way really to think about the technology. It is the technology of engineering at the level of molecules. And I think if we develop that, it becomes a sensible area, which I think is really full of promise for this. So I just will finish off on one last thing. It is the whole business of saying, well, if it doesn't occur in nature, we really need to control the scientific population, the population of people who work in laboratories, because that could fire it off. So I want to tell you a true story. I was very keen at one stage really to do a dangerous experiment, just really to see what's the limit, because, nobody knew what you had to do. If you want to contain it in a lab, we should do it dangerous. We actually did one at NIH finally, but it took years to get it going. You want to really know, can animals be infected with DNA? So, can they be infected with DNA in a plasmid? Of course, as I said, all those experiments were doomed to success. They were going to work, because the barriers as we conceived of them at the time were not as thin. So I conceived of the following experiment. I would take ricin, which is the thing the KGB used to knock off people in the London train, very poisonous material. I would get the sequence for ricin, which was known at the time, and I would clone it in lambda, and I would put it in E. coli. Little did I know at the time that nature has already done it, because the toxin of shigellosis is exactly a homologue of ricin. It is cloned in epsilon, which is a phage. Phage is defective, which I believe the successful bacteria don't want to give it to someone else, basically. So it's a defective phage, and it's in Shigella, which is a close relative of E. coli. And now that we understand so much about the evolution of genomes and know of many cases, DNA moves freely in the world. And so I will just finish by telling you that at the time when everybody was saying the recombinant scientists, it's the Pandora's box idea. If we keep it closed, you can stop it. You can kill it before it's born. That idea that controlled the scientists and you'll be safe didn't work. But in the same page of your Federal Register, there is the previous one calling for meetings on how to deal with antibiotic resistance. What caused antibiotic resistance? We know where the genes come from. We know that what that was caused by us loading into animals and huge excess of antibiotics. So selection is an important rule. And if you like, that was an environmental change. Had nothing to do. Nature provided the bits and pieces, but we provided the conditions for these to survive. So I think it is extremely important to understand about transmission, to understand about how the environment can be, in that case, unwittingly used in order to increase something which was really undesirable and threatens our control of infectious disease. So that, I think, if you put that into perspective, that you need something more than DNA, you need actually to create a public hazard, then I think that is the thing. And in conclusion, I do agree with what one of the speakers said, watch out. You need to watch for the technology that can make things not only worse, but also better. So I think, for example, those are the warning signs that I think is there.
Dr. Brenner, thank you for that summary and some provocative thoughts, as a matter of fact. I will turn now to our chair, who I believe has a no, stay put.
No,
stay put.
I have a question for you, Sidney.
Okay.
We've talked about, and we've used the phrase among the commissioners and yesterday in the public session, of responsible stewardship, of the responsibility, basically, of the human species to be stewards of the environment, of the future generations, to make sure the world has a future, and ideally a future at least as good, if not better, than the present. We've also heard a lot of people concerned about not only synthetic biology, but about genetic engineering and a lot of what the biosciences do. And I heard you say that the proof that, and I think this is a very succinct way of saying it, and leads to my question, the proof that you have the right structure is that you can synthesize it. Now, how do you address the concerns of individuals and groups who think that this ability of the human species to synthesize in order to prove things is destructive of our stewardship of nature?
Well, I think that's a question which can only be addressed by education. So I think we live in a world where increasingly people are suspicious of all technologies. And I think the only thing we can do is, I think, to educate the public. And I think it does require careful thinking about what their answer should be, and also realization. If I can give you one example, I was giving a lecture on cloning, and someone in the audience stood up and said, "Why can't I clone myself and keep the copies for spare parts?" My answer was, "Be careful, one of the copies might keep you for spare parts." You see? Now, I tell you, that is something we are guilty of. What we don't realize is that in the way we talk, we begin to view people just as genomes. I think all of this. Whereas you are much more than a genome. You are your memories, your immune system is different.
Yes.
And I think to guard against the individuality, I think that also everybody who's practiced medicine will realize that people have no notion of probability. If you say to a patient, if you take my treatment, you have a 60% of being cured, he says, am I in the 60 or the 40% club? So I think we have to address the public really as individuals. And I think we really need a way to educate people into acknowledging that the best way they can look after themselves is to look after other people.
Yeah.
So it means we must take, and I think that is one thing that can be done, because our perspective of adding all of these things has been to depersonalize the individual. But as long as we say we are dealing with persons, not just genomes or bodies, then I think it is very important to start from that point of view.
Yeah.
I think that that's just an attitude and a broad philosophical attitude that I think needs to be corrected in the way we talk about this, just the language we use. That is why I urged Drew Endy not to use the word synthetic biology. Because I said, well, one day he will be called a synthetic biologist, and that puts him in the class of plastic surgeons, animal behaviorists, and all the other fat biochemists. Yeah. I believe they call lipid biochemists. We used to call them fat biochemistry. However, but I think it's a bad choice of words. The same with genetic manipulation or modification. Should have been called genetic enhancement. They would just have carried a different kind of context to the public. So I think that when we do this, we really need to think of always identifying with what's
the terms.
outside.
So organic chemistry got it right, even though it does synthesis over and over again. Because it's called organic chemistry. It threatens nobody.
Correct. Correct. But synthesis threatens people.
The term.
The microphone is open, and also the table is open. If there are questions. Oh, okay. Go ahead.
I loved your, I guess, somewhat playful characterization of the invention that got us here last spring as a forgery.
Yeah.
I'm wondering if you were challenged to put those discoveries or those innovations in the most positive light, what language would you use? It's not a forgery, but instead it's saying, what is the real sort of
Well, I think
innovation
that you
there?
have to communicate first that the DNA is just a description of that. That in fact, in all of science, what we'd really like to do is to be able to, not to read it just as a string of letters, but to understand it. So I take the view that this is a script, like an ancient script, which I need to decode. Right. And I need to understand it, because I'll not understand evolution until I understand what is said in this. So that is the essence. Now, of course, in order to do that, what Venter has done is he has shown you can substitute one genome by another. But we have to, and that's okay. He's also shown, by the way, a much more dramatic, because in the course of this he revealed it. The big problem is, could you synthesize 1 million base pairs accurately? In fact, the thing didn't work. Firstly, because there were mistakes in the synthesis, which he had to go back and correct. So, in a technological sense, he shows it can be done. But I think when cast in the way of, we have synthesized a new life form, it isn't. It's a counterfeit of something that already exists. And it's, of course, guaranteed to work because the two things are related. But it shows in principle, or in practice, that it actually can be done and will provide a tool. Though not many people are going to have the $1 million required to synthesize the sequences.
Sidney, your line of reasoning that the DNA is this script, as an ancient script, matched to, at most, a very narrow set of readers that could do anything with it. And the two things it needs to do, presumably, is to replicate it and implement its code.
Yeah.
Seems to put more distance between where we are today and where we should be worried about.
I think so.
Well, talk to me about that.
Well, I think my own experience is that it is extremely difficult to make something new.
Yeah.
It's extremely difficult to make something new by design, intent, and so on, because we just do not understand enough about two levels of this, which is how proteins. Because what we really want to design are proteins that do the work. Because if we've given a protein, I can write down the DNA script retrospectively. Yes. It's not to sit and play with the DNA sequence, it's to play with the protein sequence. So that technology has to be developed, understood, what are the possibilities. And I think that we are very far from being able to do this. Very far, very distant. And that's just one thing. Because the other thing is, and anybody who will tell you this, that if you use other microorganisms even to express these sequences, the microorganisms don't like them. Because if it's not useful to the microorganism, they'll throw it out because they can grow faster without having this added burden. And that's very important because all attempts are made to make sure that your production strains don't lose the gene. And even deletion would help, because microorganisms are just organized to grow as fast as they can, and anything that grows even slightly faster will win out in the end. Will win out. And I think I can just make one other addition. The perspective. Half the genes we see in bacteria, we don't know what they do. But that is because many of the organisms have two lives. They actually have two genomes. They have the genome that they use to live out in the wild here. Then they have the genome that they use to live in you. or on a plant or somewhere else. Or if you take this, you find about half of it is devoted to basically being out there in the wild and just growing, and the other half is to gain from doing this. So we have lots of genes that allow bacteria to stick in you, to do things so they can actually live on the food. They get a lot of free food inside this. And, of course, some of them even decline and become essentially parasites, really dependent only. They've lost their free living character. So I think we need to have a perspective of the whole of this microbial life when we start to think it's going to get out into the environment. It's hard work surviving out there. And as the famous remark it's called the Red Queen hypothesis. It's a very famous remark that the Red Queen makes to Alice. She says, it takes all the running to stay in the same place. Because you're competing against everybody outside. So I think those perspectives need to be understood if we really are to treat this as a serious thing.
And we must. Lonnie, did you have a comment or question?
I just wanted to ask you, sir, I'm not a scientist or anything, but just
Lonnie,
to be...
use the mic.
I'm
Thank
sorry.
you. Thank you.
Just to be clear, so what you feel is that SynBio doesn't really present the danger or the urgency of danger that we have discussed and that we have been discussing, but more of a natural danger of people infecting themselves with natural pathogens and such presents a bigger danger.
Well, I think the world is dangerous as it is. And I think that things that go on simply because of the development. For example, the flu epidemic at the end of the First World War took 20 million lives, but it took nearly a year, more than a year, to go around the world. And today that is shortened 24 hours. That's all you need for an epidemic to move, and we can trace it in all the events. We have viruses now moving in at least 3 cases, and recombinant flu is one, between animals and humans. Sure. And I think that we are offering, by not changes in genetic engineering, not of the DNA, we're offering now the possibility of the way the world has developed and so on, of creating new infectious agents just by letting nature invade us, so to speak. So I think that that's the important thing. I think that what one mustn't one must avoid is to become a techno freak. A lot of that comes out of thinking about scenarios, worst case scenarios, the existentialist thing. Right. By the way, those people thinking about the nanobots that will invade the world should also start to think of could they be made accurately? Could they copy themselves accurately? When they start attacking each other, because one group might have a mutation in it. So it's the things we have to understand about complex things. Maybe I can finish off with one more true story. I was at Asilomar. One of the journalists said, the trouble with you scientists is you want to make people. So I said, well, look, I can think of much more pleasant and cheaper methods of making people than messing around with DNA in a lab. So I think that's the reality principle.
Sydney, thank you. We'll let that be the last one. Thank you very much.
Thank thank you all for being an extremely attentive and indeed impressive audience. And I want to thank my fellow commission members and Dr. Brenner once again. We are adjourned. We will reconvene in November at Emory University in Atlanta. But one final thank you to the staff of the Bioethics Commission, headed by Valerie Bonham, our executive director, for really enabling all of this to happen. Thank you so very, very much.